Each year, Microsoft Research hosts hundreds of influential speakers from around the world, including leading scientists, renowned experts in technology, book authors, and leading academics, and makes videos of these lectures freely available.
I'm trying to present to you a path that will lead to Luca Cardelli, but in a peculiar way. First of all, something happened long ago. We don't know how, but a very complex molecule got organized into a membrane. And there was LUCA, as formed by membrane, lots of proteins, and inside it, this incredibly complex molecule. Hold a second, but by LUCA now, I mean the last universal common ancestor. There is indeed a common belief that as we all share, all living beings, the same DNA, just a matter of different amount, that there is actually just one occasion in history where this happened long ago. But what happened after LUCA? There is an apparent increasing of complexity. Namely, we start from one DNA structure and membrane, some proteins, that's the original bacteria, the bacteria still as they are now, then eukaryotic cell, where there are many substructures, microtubules, there are mitochondria, chloroplast, and much more than that. You can measure, for example, a difference of complexity by counting membranes, and that's something that LUCA can do very well. And even more membranes if you go to a multicellular organism. So again, by some topological measure, you can say that there is an objective increase in complexity. This story of progression, sort of progress towards what? Well, if you look at primates, then they get more and more complex, at least the muscles of the hands, the face, the neurons, the number of neurons, the number of connections, more and more complexity. A fantastic progress till the highest product of animal evolution we got here. So we got from LUCA to LUCA, happy birthday, LUCA. Well, to be fair, it didn't work this way. No. It is not exactly this way. Well, I will try to summarize a short analysis of a marvelous work done by Stephen Jay Gould, a biologist of evolution, concerning an attempt to justify the increasing complexity. Because you see, what I said before, even though not in a linear way, in a progressive way, did happen. At the beginning, there were just a membrane with DNA, and then something got more complex. Where and how? Without aim, without a direction, without aiming to LUCA, to be honest. But how to analyze this? Then Gould, in a book which is very informal, it's a biologist book about evolution, plenty of data, observes that there is surely no bias. You see, because if you have a bias toward more complexity and you put here some sort of measure complexity and time here, all living beings would get more complex in a way or another. Well, this is the actual situation. Bacteria are still there, not more complex than the original one. Perhaps they have some more proteic activities, but basically even the simplest ones are still there. So the analysis by Gould, which is based on paleontological evidence, is that if you put some sort of complexity measure here on this line, not yet specified by, not specified very clearly by Gould, and here you put the least complexity, the bacteria, and as a value, the density of biomass, the biomass density. You have the shape like this. Still now, bacteria are about 50% of the biomass. So from this original situation, the explosion of bacteria, there's slight complexification, you go to something like this. How comes that there is a complexification? Well, this is just a random diffusion, but an asymmetric one. The basic idea, informally, that we did model in a more careful way, is that when you have an asymmetric diffusion, like an explosion of a gas against a wall, the asymmetry is propagated even by a purely Brownian motion, by just a random path that composes any diffusion. intuitively, you can understand that the Brownian motion of a particle here bumps against more particles on the left, and then is forced, on the average, to move slightly to the right. This happened in this very access space of complexity. Let's make clear one point concerning randomness, that randomness in biology is not noise. Unfortunately, there's been the dominating paradigm since long, where even evolution is seen as the result of noise. Randomness must be understood in a completely different way, because randomness implies variability, which implies diversity, and adaptation, which are a component of structural stability. In most physical theories, randomness disturbs stabilities, and then you get, by resilience, back to a viable situation. In biology, it's quite the opposite. Variability and diversity are a component of the stability of a species, of a population, even an organism. Our lungs are more stable because there is a random component in the generation of, in their morphogenesis. Similarly for the vascular systems and so on. Within an organism, within a population, within a species, again, I insist, randomness is an essential component of stability. Now, how to understand this spontaneous symmetry breaking that is at the core of the dynamics I mentioned, and also how to be precise in measuring this phenotypic complexity, anatomic complexity. You see, if you look at the lungs of a frog, they are just two containers with topological complexity. You can measure the fractality there. It's just the surface, the external surface is 2. If you go to a primate, for example, you have 2 topological structures here and here, and you can measure here the fractal dimension, which is about 3.2, and here is 2.4, because one optimizes the distribution of matter and energy of a volume within a volume. The other one optimizes the distribution of energy and matter by surface into a volume. So you can compute this, and you can say that 2.4 and 3.2 are greater than 2. So there is an increasing complexity going from one case to another. What we did some time ago, and LUCA already heard some more detailed lecture about that, but I want to go to some more recent facts, is that you can evaluate mathematically associate a number to this anatomic complexity. There is no analysis of functions. It's just anatomy of the dead animal. You can make a counting, associate a number to the differentiation. You can count the number of tissues. It's very epistemic because you decide what is a tissue, but that's rather robust. You can measure connexity and fractal structure. For example, we have 600 muscles. A cow or a horse has 400 muscles, and they say 600 is more than 400. You can measure the fractal structures, as I gave before an example. And there are also metabolic relations. You can measure networks. Neural network is a typical paradigm. We have more neurons than many other animals, but an elephant, for example, has many neurons as we have, but we have many more synaptic connections. So these are associated numbers. Being more complex doesn't mean to be better. Evolutionarily, the bacteria are the best because they're still there, and they're still the majority of the biomass. So by specifying this informal notion of complexity, we could model the process of complexification just as a random diffusion, where a diffusion is always based on random path. Technically, this is done by mimicking a Schrödinger equation, which gives the dynamics of a probability measure, while we did describe the dynamics of a potential of variability. And this is the mathematical model. What's been very surprising to me is that some leading biologists of cancer got interested in this analysis of complexity along evolution, as they see cancer as an evolutionary problem within an organism as an ecosystem. An organism is also an ecosystem. It's not only an ecosystem because it has a much stronger coherent structure than any ecosystem. Yet there is a peculiar phenomenon that is the lack of control of evolution inside an organism, that the way that these people consider cancer. And then we started discussing together, and the approach we are working now to cancer concerns the lack of control of proliferation. The main assumption we make is that selection is an essential component for the control of proliferation in an ecosystem, and that within an organism it's what keeps cells quiet. The default state is proliferation with variation, is not quiescence in an organism. This goes very much against the dominating theories, in particular in case of cancer. Our thesis is that cells in an organism are just cells, are living cells, so a priori they proliferate with variation. However, the tissue, the organs, the neural system, the immunitary system, the organism altogether controls in a differentiated way the proliferations of cells. And what happens is that in a cancer, there is a rather surprising fact. It's probably the only disease which causes that. There is an increase of complexity according to our measure. For example, topologically, this is the normal situation of a duct. This is a section of a duct. In a cancer, you have the lumina, the lumen that increases. So you can measure the increasing complexity due to uncontrolled cell proliferation. What happens is that complexity increases, but functionality decreases. And that's the problem of the pathology. So this goes on currently with a fine analysis, which also associates exact numbers to epithelial cancer, which are 80% of the known cancers. So, and it allows at least to make some sort of preliminary discrimination within this view that cancer is development that goes ori. Is this just part of an evolutionary process where a carcinogen goes in this peculiar ecosystem and disrupts the control of proliferation? So this conjecture of ours, because we are confronted with very complex empirical evidence and an incredible amount of work, I must say, because that's of course a disease which is much studied, unfortunately, because it's strongly increasing. So what's surprising is that in spite of the increasing success of therapies, therapies are fantastic now. Medicine is just, and surgery in particular is extraordinarily effective. The deaths are not decreasing. Actually, are slightly increasing. And that's because for any age group, the cancer is increasing. And is increasing largely because endocrine disruptors. Last century we produced 80,000 artificial molecules. And there is clear evidence that finite combination of these molecules are endocrine disruptors and thus indirectly carcinogens. Now, finding the finite combinations of these 80,000 molecules is not a trivial fact. Okay, I stop here. This is, in my view, a fundamental book for the analysis of the disease, which is being increasingly successful. That's my first book in biology, and that's our recent work. Doesn't discuss at all the issue of cancer, but there is a chapter on this issue of increasing complexity along evolution. Okay, happy birthday, Luca.